Hello, everyone, and welcome to today's webinar, Practical Considerations for Designing and Executing High-Content Screens. I am Christy Bach of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Thermo Fisher Scientific. To learn more, visit thermofisher.com. Before we begin, I would like to remind everyone that this event is interactive. You can submit as many questions as you would like during the presentation. To do so, simply type them into the Ask a Question box and click Submit. And with that, I'd like to now welcome our speakers for today. Dr. James Evans, CEO of PhenoVista Biosciences. Also joining us today is Steve Titus, Senior Manager of Product Management, Protein and Cell Analysis Group at Thermo Fisher Scientific. James, we'll pass it over to you to get us started.
Hi everyone, I'm James Evans. I'm CEO and founder of PhenoVista Biosciences. Thanks for joining us today. We're going to be going through a short presentation here giving practical considerations for designing and executing high-content screens or high-content assays. I'd like to thank our host, Thermo Fisher, for inviting us to give this presentation. And we'll be having a question and answer session at the end of the slides, so please stay on for that. So here's the, oops, I should press the right button here. There we go. And so this is the outline of the presentation today. So we're going to have a quick intro of what is HCS, or high content screening, and then what are its advantages over other assay strategies. And we'll have three short examples to give you a sense of sort of applications that can be tackled with high content. Then we have some practical considerations for things to bear in mind when designing a high content experiment. And then finally, about 300 or 400 slides plugging our services at PhenoVista, but that should only take a couple hours. No, it should be very quick. So slide 3 here is what is high content screening. So you'll hear the terms high content screening or HCS. You may also hear high content imaging, HDI, or even high content analysis. Essentially, they're all the same thing. And so really, the main aspects of high content are the combination of cell biology and automated quantitative imaging. Typically, that's fluorescence microscopy with a robotic autofocusing microscope that includes image analysis software. So what the main value of high content, especially in drug discovery, is its ability to really interrogate aspects of human disease or even animal disease, but to be able to make measurements of cell populations with sort of the appropriate levels of resolution. So, you know, you're not getting electron microscopy levels of resolution, but you're getting a good combination of, you know, micron level or multi-micron level, certainly the ability to discriminate major compartments within cells, and the ability to look at multiple markers at the same time, so you can multiplex. And then you've also got morphological analyses. And then this can also, and in recent years, we've seen you know, increased applications using more complex models, you know, human iPS-derived cells, 3D structures, both spheroids and organoids, microfluidic platforms. So there's a really versatile platform. And then if we go on to this slide, you can kind of see the workflow of high content. So starting off, you've got your biology, and that's actually often the most challenging part of doing high content, is getting the biology to perform as expected and to work in multi-well plates, and for the you know, all the analysis and also all the staining and the control treatments to work as expected. So yeah, so you get the biology right, you've got the liquid handling for treatments and you know, culturing, And then you've got the sort of the key part on the sort of technology side, which is the automated microscopy. This can be done live and fixed. Often, a lot of the systems right now use spinning disk confocal, which is a good compromise between resolution and rejection of out of focus, blurry light, and speed. And magnifications can range from, if you're looking at a whole organism or large spheroids, really low magnification, 1 or 2.5x. 100x is possible, though quite rare. I would say the majority of high content assays are done in the sort of 10, 20, or 40x magnification range. And as we mentioned before, this can be done in 2D or 3D. Once you've got all your images, you need to get numbers from those images, and that's where the algorithm is designed to do to break the images into regions of interest and quantify objects within the images, like nuclei, shape, intensity of your marker of interest. And then you've got loads and loads of numbers, and that is where, recently, the sort of big resurgence of AI and machine learning, being able to mine out trends that might not be immediately apparent to a user comes in. So, what are the advantages of high content over other assay strategies? We'll go through a little bit of that. So I would say the main advantages of high content is in the name. You get lots of content, you get lots of information. It can be a good thing and a bad thing. Sometimes it can be overwhelming, but with the tools available today, it's a lot easier to mine through that data. But really being able to combine some of the sort of commonly used traditional methods like Western blots and ELISAs and things like that to quantify the amount of a protein that might be in a population of cells. You get that with imaging. You can look at expression of your target in the cells, and then what I think the real key value is the ability to know not only how much protein is being made by your cells in the population, but where is that protein, both subcellularly, but also within the population. What is often a surprise for people who are not familiar with this type of approach is all cells are not the same. You can see just on this example on the right here that the cells have a distribution or a variety of different shapes, different staining intensities, even their nuclei vary slightly. And this is all part of this sort of the natural amount of variation that you get in biological models. So being able to quantify that is really important. And then with high content, you can also do fixed time points. But you can also do live imaging too, and get sort of continual measurements of the biology over time. And of course, that would include measuring things like the viability of the cells as well. So yeah, I mean, the things that you can't get from a Western blot, single cell data, subpopulation data, combining both the morphological and pathway-specific readouts with the viability of the cells, and then dynamic and morphological changes. So high content has a lot of advantages. So we're going to go into a couple of examples to hopefully illustrate those points now. First one is an example using lung fibrosis, where we combine quantification of protein expression, the health of the cells, and morphological changes, and actually the co-localization of a couple of markers of interest. So fibrosis, if you're not familiar with it, is a really prevalent disease that affects lots of aspects of biology, but essentially is a repair mechanism that's gone kind of overboard and leads to tissue stiffening and can lead to organ failure. Interestingly, it's also implicated in cancer biology too, where you can get a a sort of a coating of tumors in a fibrous network that then makes it difficult for treatment to occur. So here's a typical example of how the high content images look. So here, going left to right, we've got Hertz, which is a common stain that we use to label the nucleides in nearly every assay that we run. This allows you to demark where the cells are relative to the background of the image where there are no cells. In this case, in the second column, we're staining for a assay-specific marker for alpha smooth muscle actin. And then in the right, the next one over, and with the red label there is F-actin. This is typically labeled using phalloidin, which is a really great dye that's used a lot to label filamentous actin. And then the furthest right column there is the composite, where we overlay the pseudo-colored images to kind of see where all the markers are relative to each other. And going top to bottom here, you've got the untreated cells who are sort of non-fibrotic. And then in the middle, induction of fibrosis with TGF-beta induces alpha SMA, induces F-actin. The stress fibers are upregulated there. And then on the bottom, we co-treat with TGF-beta and an inhibitor, ALK5I, and you can see that suppressed the activation of fibrosis. So this gives us what we call our assay window. So here we can kind of drive the behavior of the biology in two directions, and then screen unknown test articles to see where they lie between those two extremes. And so this is using human lung fibroblast primary cells. And so how do so those are the images, but we actually have to get numbers from those images. And so on the left here, on the right, you can see the what we call the masks. So these are where the algorithm goes through and quantifies the pixel intensities and the shapes and the amount of the area that's occupied by a specific stain. In this case, the ones that are of most of interest to us are alpha SMA and F-actin. So you can see the pink masks on the top and the yellow masks for F-actin below that. So we can define the fibers in each of the channels. And then click the wrong button. So then all this can be output numerically. And on the right-hand side of the top there, you can see the average alpha smooth muscle actin fiber area that increases with TGF-beta. And then on the right side of that, when you co-administer the inhibitor ALK5I, you see that increase in alpha smooth muscle actin does not occur. Similarly, you can look at F-actin fiber area which is plotted below that. You see a similar trend. And so, one of the other really important aspects, because we take an imaging approach, not only do you get abundance through intensity of the markers, but you can also look at the organization of the fibers and get some additional information. So using a fiber alignment metric, we're able to measure the organization and essentially how the fibers in each cell are running parallel to each other, and that can give us an additional readout. So doing things like that is not possible unless you're doing imaging. So again, that can be plotted here in the top right there. You've got F-actin fiber alignment increasing with TGF-beta administration, being suppressed with the presence of the inhibitor, and similarly with the alpha smooth muscle actin fiber. So it's a similar thing, though a little bit noisier. And in this, so here we're looking at the co-occurrence of alpha smooth muscle actin and F-actin. So so we can see on the top plot there, with increasing amounts of TGF-beta, you get an increase in the overlap between those two signals. And then when you treat with the inhibitor, you can see that that increase is suppressed quite nicely. And I think here's another slide here. Oh, so this again, this is really speaking to the high content aspect. There's a lot of different aspects of the data that you get, from a study like this. So here we're just looking at the cell shape. So we can measure changes in the cell shape that occur with administration of TGF-beta, the induction of fibrosis. So you can see in the top plot there, we use this metric called perimeter to area ratio, which draws a line around the cell and then calculates its area within that boundary. And you can see that perimeter to area ratio is diminished in the presence of TGF-beta, and there's a similar. There are lots of different ways to measure cell shape, lots of different metrics. Things like convex hull area ratio is another one that's quite popular too. But here, length width ratio is one that can similarly be used and shows a reduction that can be suppressed by blocking TGF-beta with ALK5I. So, in summary, using a lung fibrosis, looking at lung fibrosis, and I should mention that there are similar approaches that can be applied to liver fibrosis, cardiac fibrosis, and several other dermal fibrosis, for example. But the main thing here is that you get lots of different readouts that can provide scientists with clues as to how their test articles might be working to suppress the disease. And this really gives them a way to differentially rank how well their drugs that are in development are working, and provide them with a competitive advantage over other drug companies on the market. The next example is, I guess, similar in some ways, in that it's sort of related to sort of inflammation gone wrong and repair mechanisms gone wrong. But here we're looking at, in the CNS, looking at microglial phagocytosis. So microglia, for those who aren't familiar, are essentially an immune component of the CNS and the brain. And again, when they're behaving, it's all good. When they go overboard, it can create inflammation and create symptoms of disease. So one of the things that we can do is use recently available human iPS-derived microglia to measure how well they're performing their primary job, which is phagocytosis, or ingestion of foreign particles. So this assay, in a different way from the previous assay we just talked about, is actually a live cell assay where we use a time course and make measurements at sort of a relatively high frequency. So you can see that on the left plot there, that you've got multiple measurements made over a 24-hour period, and you can compare the rates of the increase in those lines with the different treatments, stimulating with IL-10 or suppressing with cytoclasm D, which blocks actin polymerization, and then you've got the controls on there too. So how we do this is we use a pH-sensitive dye called Frodo, which has been conjugated in this case to bacterial fragments, which the cells will ingest, and then when it gets inside the cell and goes to a acidic compartment, it fluoresces. So that's how we can measure that over time. And actually, I should mention there also we can suppress the phagocytosis not just with cytoclasm D, which is a sort of a sledgehammer negative control that sort of shuts down the cytoskeleton of the cell, but we can also suppress phagocytosis with interferon gamma, which is a sort of more physiologically relevant input to slow down the rate of phagocytosis, which you can see there in the orange trace. And here we have another slide. I guess that just summarizes there that you can not only see the trends in the data, but you can also, compare and make sure that the images substantiate the data that you're getting, and that you have a good match between what your eyes are seeing in the image and what the numbers are being reported as. So for this assay, until recently, probably 4 years ago, 3 years ago, human iPS-derived microglia were not really commercially available. So researchers often had to use less relevant models like the mouse BV2 cell line. But yeah, now that we can use this human line, we're able to measure not just the rate of phagocytosis, but also the sort of maximum capacity. So you can see whether those traces for phagocytic activity, whether they maybe start off at the same rate, but they max out and the cells get exhausted or something like that. So there's a lot of information that you can derive from that. And then importantly, there are follow-up assays that can be performed where you can increase the complexity. For example, co-culturing the microglia with neurons and astrocytes and look at the interplay between those 3 cell types. It's something you can do with high content too. Okay, a final example is something called cell painting. This is a really popular approach now that came out of the Broad Institute, a former colleague of mine, and Carpenter. Really came from the need to quantify effects on cells that were being used in siRNA screens, and they needed a very unbiased way to label the cells and determine, if you knock down a gene, what was the effect on cell viability, maybe on the mitochondria, not knowing what the effect might be at all. So essentially, cell painting uses a combination of relatively cheap, easy to apply dyes that demark the major compartments of cells. And like I said, this reduces the labor involved with sort of doing hundreds and hundreds of plates. If you want to do that, it makes it a lot easier to scale. It also is lower cost than if you were using antibodies, and primary and secondary antibodies, and which are also more time consuming to apply. But most importantly, it doesn't really bias the data to a specific pathway. It's really looking at the major compartments, the nucleus, the ER, nucleoli, F-actin, Golgi, and membrane and the mitochondria. So really the major compartments. Commonly, cell painting is used in A549 and U2S cells. We see a lot of our clients in the drug discovery space wanting to take a similar approach, but applying it to sort of more tissue-specific cell types, hepatocytes, cardiomyocytes, neurons, microglia, astrocytes, all human iPS-derived typically. So one of the major applications that people use this for is creating fingerprints so they can categorize the mechanism of action for known drugs and then match unknown entities to those known, well annotated examples. So this can do two things. It can tell a scientist that their test article that they're developing or the drug that they're developing has a similar, potentially similar mechanism of action to something that's already been FDA approved, for example. Or if you're looking at a kinase library, you could say you can figure out that your drug candidate is maybe targeting the kinase that you'd like it to, but it maybe also has some off-target effects. So that can be important information too. So lots of different ways. I'm sure there's going to be new applications of it as well in the coming years. Here's how the data looks. So you've got these different labels lighting up different parts of the cell. Moving left to right here, you've got the hooks and the phalloidin in blue and red. On the bottom, you've got masks that identify the nuclear boundary and the nucleoli within each nucleus. And then moving left to right, still you've got the endoplasmic reticulum, same with concanavalin A, a fluorescent conjugate of concanavalin A. And then you've got mitotractor, which lights up the mitochondria. And then similar to the lung fibrosis assay we talked about earlier, use phalloidin, a fluorescently conjugated phalloidin that integrates into the F-actin structures, which are then detected with that mask shown in pink in the bottom right corner. Couple of examples here. This is actually using human iPS-derived hepatocytes from Fujifilm, and just zooming in here, you can see the change top to bottom between the vehicle control and the cytoclasin D treated in the bottom. So you can see that there's a whole bunch of things that have changed in the appearance of the cells, and this can all be quantified. And one of the ways, there's lots of different ways to mine through this data. So there's two examples shown here, left and right. On the left, we've got a UMAP analysis projection. That's the word I'm looking for. UMAP projection of all the test articles, and essentially this is kind of a more modern way of doing clustering, but essentially puts uses the multivariate output. So it could be 4,000 measurements performed on each cell, and so it gives you a very intricate fingerprint, which then can be used to put similar compounds with similar fingerprints in the same bucket. And then another approach, you can use one of these like dendrogram type plots here to do a kind of a ranking. So you can then rank compounds and put the similar compounds. It's essentially a similar approach, but gives you kind of two different views, and people have their preferences. But there's hundreds of different ways to mine through this data. So yeah, as I mentioned in the introduction there on cell painting, you can look into mechanism of action, target engagement, off-target effects, and then excitingly, you can also take this approach into potentially more relevant cell types too, depending on your area of interest. So finally, got a couple minutes here just to go through some examples of things to consider when designing an HCS experiment. So this again, as a refresher, is the workflow going left to right from the biology through the automated image acquisition, analysis, and data analysis. One of the very first things to decide is, what models are you going to consider? Do you think this needs to be a live cell assay or a fixed assay? There's implications. If you go with a live assay, you typically don't have as much versatility, or it's more difficult to detect specific pathways of interest, and often they can tie up your instrument for longer periods of time than a fixed endpoint assay, where you can make all the plates and then scan them at your convenience. So that's one of the major things. Also, deciding whether you're going to go with a 2D model or a 3D model, if you're going to use microwells, so standard microwells, or use microfluidics, bioprinting. There's lots of different options. So regarding cells, we use a lot of iPS-derived cells. They have a good combination of biological relevance and consistency. Primary cells are great, but you do have to worry about batch-to-batch variation. Typically, in sort of when high content was first developed, so 25 years ago, there's a lot of cell lines. Since then, immortalized cells have really been used less and less as the least relevant, I guess, and physiologically relevant. And then, of course, you can use patient-derived samples too, so you have access to those. As I mentioned just a couple of slides ago, there's also the design consideration around whether you use a 2D approach, 3D, bioprinting, microfluidic. Essentially, it becomes a trade-off between throughput, cost and throughput, and sort of complexity. So with 2D being the sort of the easiest, the highest throughput, and the lowest cost, and some of the bioprinted, microfluidic or 3D approaches having various degrees of cost and complexity, but hopefully more relevant data. Controls are absolutely key. Getting the biology to perform as expected, as it matches with the literature, and that's really going to create the sensitivity, reproducibility of your assay, and the ability to rank input treatments relative to each other. So yeah, that's probably one of the most important aspects of getting not just high content assays, but any assay to work, is really the getting the assay window to be as linear and as large and reproducible as possible. So yeah, there's often a strategic decision to be made at the beginning of your assay, whether you want to have something that's very unbiased, like the cell painting approach, or you want to have something that's very targeted, like the fibrosis approach, where we're looking at specific aspects of biology. In reality, you can often combine those approaches and have some amount of unbiased aspects that look at fundamental aspects of biology, like the lysosomal degradation, and then that can be combined with a target of interest through use of an antibody. So, of course, you can run two assays at the same time too. So there's a lot of flexibility there. Yep, dyes and antibodies. Dyes are great, a lot easier to use than antibodies, but there's far fewer dyes are available compared with antibodies. Typically, most systems are 4-channel, going from Herx in blue through to deep red and 647. Thermo have their systems, which we use as part of our offering, along with other manufacturers. They actually have a near IR, a 785 channel as well, which is a nice additional capability. And then, yeah, actually, they also have, Thermo has a nice spectral viewer tool that we tend to look at when we're trying to compare which dyes we should be using for particular assays. That's a nice tool I'll direct you to. And then scanner, yeah. So there are, so as mentioned, Thermo has is one of the major high content systems available. There are other ones out there too, and each of them have their sort of strengths and weaknesses, but they're pretty similar to each other, to be honest. It really comes down to sort of what kind of workflow you're trying to support, and your sometimes the resources that you have within your company as far as downstream bioinformatics capabilities and things like that. And then once you've got your images, some of the imaging systems come bundled with image analysis. Other ones expect you to handle the storage, organization, analysis of the images yourself, which for some people that's their preference. So again, that's an important thing to bear in mind when selecting your system and the approach you want to take. And then, once you have your assay working, figuring out how robust it is. Z-score is the classic assay method to assess whether you're getting a reproducible assay. There's also things like SSMD that we use a lot to look at the statistical significance of your assay and how reproducible it is. That's a key point that should be used to sort of measure your success in developing an assay. And then, if not all assays need to be scaled to run hundreds of plates a day, but some do, and if they do, integration with liquid handling for sample prep and for integration of the sample preparation and getting those plates onto the imager and then back off again. That's a whole industry in itself, and it's definitely important when you start getting some of these high content assays working at scale. The last couple of slides here just tell you about what we do at Finavista. So previous slides, what we do at Finavista is combine human cell biology with high content analysis to answer questions for our clients that are in the drug discovery space. Figuring out what their study goals are and what the outputs they're interested in, helping them select the models and formats that have the best chance of answering those questions, and then putting together a project and a study design with timelines and cost estimates. Two major areas that we tend to support: neurobiology, immuno-oncology, and oncology, and fibrosis, as we saw an example of. All these areas of biology really have visual components to them that benefit from application of high content. Those can include subcellular localization, target dynamics, uptake, for example, in cell and gene therapy, seeing whether a cargo protein or a gene is delivered and the protein is expressed as expected. And then I should also mention that while we do focus on high content, we do also support other measurements, both internally and through partners of ours, so we can get the right kind of information from these biological models that we've helped to get working in the lab. And finally, the 4 types of service offerings that we have. So if you have images that you need analyzing, we can support that. If you have plates that are fixed and stained and you want them scanned and analyzed, we can handle that. That's a very easy way for us to support you getting the data you need. We also have off-the-shelf, pre-configured assays for things like neural outgrowth, mitochondrial health. Those are details of those are on our website. And then, as I mentioned, you saw examples of cell painting. We can support that in a variety of different cell types. And then, actually, the bulk of the work that we do for our clients falls into this custom or bespoke bucket where a client comes to us with a feasibility project, and we develop that assay and run it in a completely flexible and customized fashion for them. And so that is the end of the presentation. Thank you for attending, and we're going to be having a question and answer session next. I'll hand it back to the moderator. Thank you.
Well, thank you so much, James, for your informative presentation. We will now hear from Steve Titus as he walks you through planning your cell painting experiment using the free web-based tool StainIt Cell Staining Simulator. So Steve, we'll pass it over to you.
Thank you, Christy and Dr. Evans. My name is Steve Titus, and I work at Thermo Fisher Scientific as a product manager for imaging reagents. I'd like to introduce you to a recently released tool called StainIt. StainIt is a web-based all-in-one tool that allows you to plan, visualize, and order your imaging reagents via a series of guided steps. I like to think of this as the ultimate tool for a DIY cell paint or kit builder for cellular imaging experiments. Let's walk through this briefly. When you open the page, you will see three windows. The cell pane shows you a depiction of what the cell will look like when the stains are applied. The spectra pane will show you the fluorescent spectra as excited by the light source, and the structures pane is where you'll make your selections. You typically start in the structures pane and make selections by progressing from left to right. Let's get started building a kit. So you start with the cell preparation window by selecting live, fixed, or fixed retained. Live refers to products that are suitable for live cell experiments. Fixed refers to products that are added to cells after fixation, and fixable refers to products that are added to live cells that retain their fluorescence properties after undergoing fixation. So for this example, I will choose fixed. Then I will select add new stain. And I will start by selecting a structure. In this case, I'll start with nucleus. And from here, you have two options. You have a dye reporter or antibody selection. And this is where it gets interesting and fun. So you have the option to select your emission channel first, or if you know a product type that you're looking for, you can either type it in here or click in the dropdown. And in my case, I'm going to select Herxt 33342, and that automatically fills the emission channel. And you can see now the stain being applied to the nucleus, and you can see the spectra being applied over here as well. I'll try another one. I'll add a cytoplasmic stain. Try another dye. In this case, I want to do GFP. So if I click here, most frequently used products again. In this case, there's one product that's available that's fixed. You can see it being applied and your two colors. Add a third stain. Let's go with plasma membrane. Try this again. In this case, I want side 3. You can select weak germ agglutinin, or you can actually start typing as well. And if something is available, it will auto-populate. Try that. And then let's do one more. Let's do tubulin. And as an example, I'll show you when there are no products available. You see here, no products available. So let's move it over to an antibody. We'll switch to Cy5, and here's where you have your antibody options. So in this case, I will select beta-3 tubulin. Okay. When this is all done, you can see your virtually simulated stained cell. You can see the spectra here. You can review and check out. And so what's neat here is you've got all of your products available: nucleus, cytoplasm, plasma membrane, tubulin. You may also need products down here, such as fixatives, perms, and things like that. You can add all these products to the cart here and check out. You can download this as a PDF. You can download it as an Excel to share with your lab mates or put in your ELN. A couple other things of note is that you have a link to the Spectra Viewer here, which takes you to the full Spectra Viewer site where you can add other reagents. We have a user guide, And to take you back to the cell viewer, some advanced features, we have the ability to embed instruments. So currently we use the simulated instrument that has 8 channels, and this allows you to look at all products that are available. But if you have a 4-color instrument or an instrument with less colors, you can select that here. So for instance, if I select the CX7 Laser Pro, laser confocal, you can now see the laser lines that are available with this system. And so in this library, we have several instruments from different companies. And we also have the ability to make a new instrument here, or you can view and edit the configuration. So the complexity gets pretty deep, which is pretty nice. As you use this more, I think you'll appreciate the complexities. I typically stick with the 8-channel stimulated instrument in order to view the most product options, but you do have possibilities here as well. I think that's what I've got. So let's open it up for live Q&A. Thanks so much.
All right. Well, webinar. So to our audience, if you have a question that you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen, and we'll answer as many of your questions as we have time for. All right, now before we get started with our audience questions, we do have a few poll questions for you. So we have this first poll question here we'd like you to participate in. If you don't mind seeing this question here, would you like to add, would like to add this many markers to my experiment? 2 plus hoaxed, 3 plus hoaxed, or 4 or more plus hoaxed? And you can select your answer there. We'll give you a moment to do this. All right. We'll give you a few more seconds here. Okay, great. Now we'll move on to our next question. This question asks, my primary area of interest or research is oncology or immuno-oncology, neurobiology or CNS, and cell or gene therapy. And the question asks, my primary area of interest or research is oncology or immuno-oncology, neurobiology or CNS, or cell and gene therapy. And we'll give you a few more seconds here and move on to our final question. All right, great. Well, thank you for your participation. We have one more question for you guys. All right, and this next question asks, which part of HCS in which I need the most advice or support is assay design, microscopy or imaging, or image analysis? Again, the question, a part of HCS in which I need the most advice or support is assay design, microscopy or imaging, or image analysis. I'll give you just a few more moments to submit your answers there, then we'll dive right into our Q&A. Great. All right. Well, thank you so much for your participation. Let's get started with our Q&A. So our first question here is for James, and this question asks, Does PhenoVista only perform imaging-based assays, or do you offer other types of readouts as well?
Oh, thanks. So yeah, we started our business doing imaging, but over the years we've added additional supporting measurement methods, looking at gene expression through qPCR, looking at calcium transients using FLIPR. So we do support other types of non-imaging based approaches.
Great, thank you. All right, Steve, I've got a question for you. Is there a way I can enter a custom configuration for my microscope? And how would I know what configuration is for my microscope?
A great question. And let me see if I can share my screen and show you. One second. Can you see my screen okay?
Yes, we can.
Okay, perfect. So real quickly, you would go to the instrument settings here. So you start with cell, go to instrument. We have several that are pre-populated. So we have Thermo Fisher systems, PerkinElmer systems, Sartorius, Yokogawa. And if you have others that you're interested in, please feel free to reach out and we can add those as instruments in the software. You also have the ability to make a new instrument here, and you basically follow the prompts. So you can say custom, or you could actually make an instrument based on a pre-populated series of settings. But essentially, you have the ability to do that here. And there's many things that you can do. You can save these settings and import them to other sessions and use those for your experiments. So absolutely. Thank you.
Great, thank you so much, Steve, for that visual as well. All right, another question here. I believe this is going back to James. What kind of assays has PhenoVista done in the neurodegenerative space? And what kinds of multicultural models have you developed? And the third part of this question, what kinds of readouts can you look at that are relevant hallmarks of disease?
Yeah, that's a great question. Yeah, when we started the company now 10 years ago, Tony and I really focused on iPS-derived neurons as a relatively new product that we wanted to help get in the hands of scientists or the data that you could get from them. So yeah, neurobiology has always been a great fit for high content because you unlike maybe immunology where you can use flow cytometry, in neurobiology you don't have that luxury. So yeah, to this day we more than half of the studies that we do for clients is in the neurobiology space, and those will be as simple as neuro outgrowth. Can get more complex version of that where we look at synapse formation in longer term cultures. And then to your question about multicultural systems, so combining astrocytes, so human iPS-derived astrocytes, particularly with different types of neurons, really helps their maturation process and makes the data more relevant. You can actually go now one step further and add in a third component like microglia and look at the neuroinflammation processes. And these can be scaled from 2D to 3D as well, where you can do sort of calcium transients, look at excitatory events using FLIPR-based systems. And then in terms of that, there would be a relevant readout for function. And then going back to sort of high content approaches, so you can use dyes to look at things like mitochondrial health, and any antibody really to a particular pathway that might be relevant, but also proteins that are implicated in neurodegeneration, like TDP-43, for example, and phosphorylated versions of that. So you can look at the abundance, the formation of aggregates, and the location of those aggregates. So yeah, really a really active area for us at Finavista.
Great, thank you. All right, we've got some more questions coming in here and a bit more time, so we'll continue with a few more before we wrap up. This next question asks, when imaging 3D samples, do you typically collect several Z sections or just a few?
This question for me again. It really depends on the biology and the throughput requirements. So, if it's a relatively small study and you've got, time and the biological samples are relatively thick, and by relatively thick, that could be a few hundred microns in Z depth, then, you might have the luxury of taking as many Z slices as you want, which might be a couple hundred, but that's not typical for high content. Typically, in high content approaches, you have hundreds of samples that you want to get through, and more often than not, they're usually on the thinner side rather than being hundreds of microns thick. That's not when you're looking at organoids, those can be thicker. So this is the trade-off between throughput and sampling in 3D is something that we developed during the assay development validation approach or stage to figure out how many slices do you need to sufficiently survey the samples and get the data quality you need. And so that'll be dependent on what those what that signal is that you're looking for and how dispersed or regionalized that signal is in the 3D space. And so to answer the question, it depends, but typically it's a few slices, so it could be anywhere in sort of the 3 to 10 slices is pretty typical.
Great, thank you. All right, next question here. How important is it to consider cell density as a confound when analyzing the effect of perturbations on your phenotype of interest?
Yeah, it's hugely important. Cell density, will affect morphology, will affect how sort of comfortable or resilient to stress the cells can be. And then it can also impact sort of less relevant but still important aspects. So, as the cells maybe get too dense, you start having cells detach in some cases or forming, 3D structures that make imaging more challenging. So, cell density is one of the very first aspects of assay optimization that our scientists will tackle.
Great, thank you. All right, another question for you, James. What kinds of assays have you run in the immuno-oncology space? Any T-cell killing?
Oh, yeah. So, as I mentioned, when we started Finavista, it was really neuro-focused. And a few years after we started the company, the CAR T field really took off. And, Tony and his team in the lab, worked hard to support immune cell analysis, which is not typically well suited to imaging. It's more cells are typically weakly adherent and often in suspension, and we worked hard to develop assays that would be relevant to people in that field. And one of them straight off the bat was solid tumor cell line killing. So culture the cells in 2D, the target cells, typically a tumor cell line, and then take primary T cells that have been isolated through flow cytometry and apply those and look for killing and interactions between the T cells and the target cells. And that's a really powerful way to assess the effects of T cells on specific types of tumor types. And then going beyond that, you can also take that into a 3D context where the tumor is grown into a spheroid, and then T cells or other immune components can be added, and you can look for not just killing but also penetrance, which is an important aspect. As I mentioned, when talking about fibrosis, often there is a layer around a tumor that can be sort of recalcitrant to immune cell penetration, and so you want to be able to look at that and quantify penetrance. So those are a couple of the assays that we do in that space.
Great. Well, thank you so much, James, for that answer. And unfortunately, we are out of time for today, so that will bring us to our conclusion. But I do want to thank you again, James and Steve, for joining us today for your important research. And I would also like to thank LabRoots and our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today. Questions submitted during the live broadcast and those submitted during the on-demand period will be addressed by our speakers via the contact information you provided at the time of registration. This webcast can be viewed on demand, and LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. So until next time, take care, everyone. Goodbye.
Thank you.